Welcome to Educon. In this session, we will learn about endomembrane system in eukaryotic cells. Endomembrane system. In endomembrane system, we have four organelles which are where their functions are coordinated. One is endoplasmic reticulum, another is Golgi complex, lysosomes and vacuoles. In endomembrane system, why we are not discussing other organelles such as mitochondria, chloroplast and all things? Because in function all these four organelles are coordinated. Hence, in endomembrane system we are not discussing any other organelles. Endoplasmic reticulum. What is endoplasmic reticulum? It is a network or reticulum of tiny tubular structures present in cytoplasm. So, and it divides the intracellular space into two compartments. One is luminal, which is the space present inside the endoplasmic reticulum, and the outside is the extraluminal, that is cytoplasm. And in endoplasmic reticulum, if they have ribosomes attached on its surface, then it is called rough endoplasmic reticulum. Whereas, if there is no any ribosomes attached to the endoplasmic reticulum, then it is called as a smooth endoplasmic reticulum. In rough endoplasmic reticulum, the protein synthesis occurs because ribosomes are the organelles which are involved in protein synthesis. So, rough endoplasmic reticulum, it is a site for protein synthesis and also secretion. Rough endoplasmic reticulum is a site for protein synthesis and also secretion. Whereas smooth endoplasmic reticulum, they are involved in lipid synthesis. Lipid synthesis, because they do not have any ribosomes attached to it, because ribosomes are solely for the protein synthesis in both prokaryotes and also in eukaryotes. And these endoplasmic reticulum, it will be they are extensive and also continuous in the outer membrane of the nucleus. For example, this is the nucleus. So, this is the nuclear envelope, this is the nuclear membrane, and this is the nucleus, and here they are in this form. So, they are in continuous with the nuclear membrane. Golgi complex or Golgi apparatus. It was discovered by a scientist, Italian scientist. His name is Camillo Golgi in 1898. So, it was named after him as Golgi bodies. These are Golgi bodies are they are flat disc shaped sacs which are arranged near the nucleus, and they have many cisternae here. And Golgi complex is having two phases. One is the cis phase, which is also called as forming phase, another is a maturing phase, also called as trans phase. Here, as I said before, in an endomembrane system, there are four organelles where their functions are interconnected. So, now we will see what is the main function of Golgi complex. The main function of Golgi complex is packaging material. So, these materials, from where it will come? It will come from endoplasmic reticulum, because on endoplasmic reticulum we have ribosome which synthesizes proteins which are released and accepted at cis phase of the or forming phase of the Golgi complex. Here you are seeing a materials which has to be packed. So these materials once reaches the cis phase, it fuses in the cis phase and it moves along the cisternae and reaches to the maturing phase before being released. So but the cisternae, all the from cis phase to maturing phase, these two phases function is different completely, but they are interconnected. So materials from ER fuses with the cis phase and move towards the trans phase. From there they are released into the cytoplasm. So we understand the role of Golgi complex. So what will Golgi complex do in the cell? It packages material received from the endoplasmic reticulum. For example, proteins synthesized by ribosomes on endoplasmic reticulum are modified in the cisternae of the Golgi complex before release from the transphase of the Golgi complex. And also Golgi complex is also important site for formation of glycoproteins and glycolipids. So glycoproteins and glycolipids are synthesized in Golgi complex. Lysosomes. Lysosomes are also part of endomembrane system. Lysosomes are membrane bound vesicles which are formed by the process of packaging in Golgi complex. Lysosomes, they are membrane bound vesicles. Before we discuss about Golgi complex, how the material which are released from endoplasmic reticulum and then repacked in Golgi complex and released, and those released vesicles are called lysosomes. And many isolated lysosomes are present in the cytoplasm which are found to be rich in almost all hydrolytic enzymes such as lipases, proteases and carbohydrates. All these enzymes, they help in digesting carbohydrates, proteins and lipids in cell. Apart from lysosomes, we have another organelle which comes under endomembrane system. Those are vacuoles. Vacuoles are membrane bound vesicles present in cytoplasm. They in vacuoles we found sap, water and waste product which is not useful for cell, because vacuoles they take all the waste material from the cell that are not required by the cell, because sometimes the material may be harmful for the cellular activity. It is bound by a single membrane called tonoplast. So in plants 90% of the volume is occupied by vacuole, and in animals it is different. And vacuoles can be different types: sap vacuole, contractile vacuole and food vacuole. Sap vacuole means it carried waste or any other product which are not essential for the cellular functions. Contractile vacuole in amoeba, it helps in osmoregulation in amoeba, and also food vacuoles in protists.